A long time ago, this man decided to create a place where great biology could be done and where scientists could create new, never-before-seen experiments. He called this place Biology Stadium. To fill his biology stadium, he chose the greatest scientists, each renowned for their particular style of science. He called them Iron Biologists. Iron Biologist, Anthropology. Iron Biologist, Tadpole. Iron Chef, Forensics. Iron Biologist, Intern. Today's secret ingredient is cellulose acetate. Now what is she doing with the cellulose acetate? It looks like she's getting ready to soak it. See, she's got the tub filled, and she's putting it in the buffer solution. Yes, I just spoke with the Iron Biologist. She says that to soak the gel, you must put it in the buffer very slowly to avoid getting air bubbles. She's moving a bit faster now. Her hands seem quick and excited. It seems like there's a sense of urgency. You can almost sense it in her eyebrows. Well, I think she's lining up tubes in some packed ice, actually. I think it's some packed ice that's in a styrofoam square box. I wonder why. I guess she must be lining them up to keep them cold for some sort of reason. What does she have? I think they might be snails. I can't tell for sure, but they're quite small. Let's see if we can zoom in. Oh, yes, it looks like a snail. Yes, these are very expensive snails, and they must be shipped in especially from New Zealand, from Lake Maparika, I believe. It's very sophisticated. Now she's taking with the tweezers and she's filling them up. They must need to stay cold for some sort of reason. I think I read somewhere that the snails need to be cold so that the proteins in the snails and the material that it doesn't break down. Oh, she's going to the fridge. She's going to the fridge. She's got something. It's in a bottle. It looks purple. Slightly purple. She's going to her pipette. She's setting it. Specific amount. She cracks the tip. She's got a new tip. Fresh tip. And she is taking, that must be the buffer solution. That must be the buffer solution, which she must be adding to each of the tubes in which she put the snails. I see. That must be some type of crushing buffer going into each of the tubes. Let's see how fast and deftly she moves. Did you notice how deftly she moves? Ooh. She's twisting. It's in her hand. She's wiping the tip. Yes, I think that buffer was in an effort to stabilize the proteins and to keep them from denaturing while she grinds them. It looks like she's doing the grinding now, actually. She's twirling, yes, she's grinding each of the tubes in the tube. She's grinding. I see. Getting all of the proteins from the snail mixed up in an even fashion. She's going to her pipette again. A new tip. It looks like she's She's moving the ice tray. Looks like she's done. She's going for the well. It looks like the well plate. She's just pipetted some of the snail in crushing buffer that's been crushed into the well plate. Very precise. Very precise. Notice she has the well plate on the counter. I believe she can. It's also helpful that it may be possible to put the well plate on ice as well to keep it extra cool, to keep proteins from denaturing. Because protein denaturing is not what you want. You want the full proteins. She's gonna have to work faster to get this done within the time limit. She's got the stamper out. She's going to the fridge. She's got her buffer. She's got the cellulose acetate gel. Notice how it's in the cellulose acetate gel well thing that you pour the buffer into, that see it's soaked thing, that she's pulling it out slowly as to avoid gathering any air bubbles and losing any of the cellulose acetate. Now you see she's got blot paper to dry the gel by pressing it, not wiping it, so as to not smear it or cause any irregularities in the surface, so as to impair the ability of the proteins to run smoothly on the gel. Notice she moves it up against the plate. She's stamping her wells, tapping it, jiggling it just a bit, getting a good firm stamp, moving over to the plate, stamping it once firmly, moving back over. She stamps again. She's very, very smooth, very precise. Notice how the gloves she's wearing keep out contamination. She's got that stamp. There's one lane. We see one lane right there. You can see very closely how it's up against there. I think there's some particular lines that she lines up the gel itself with. Oh, now she's preparing the gel plate. She looks like she put some buffer on the piece of glass. She's lining it up. She's getting it square. She's going to the fridge. It looks like we're going to run the gel. She goes down the gel rig. She's taking off the top. She's being careful to notice that the wick. She's placing the red side. She's placing the gel oriented towards the direction that she wants her proteins to run towards, which would be the red, which would be the positive electrode in the gel rig. She's checking her wires. She's making sure they fit. She turns it on. It's good. She's good. She goes to her timer. She sets her timer for the correct amount of time. There's the timer. We can almost see how much time. How much time does it say? She's 13, 14, 15. She's raising it up. We're almost there. It looks like this gel is going to run for 35 minutes.